All right, I guess we can get started. I rearranged the schedule a little bit because I don't think we're going to finish up with our DNA sequencing stuff today. I think it'll take us next class period, so we're not going to start Python until after the first test. So you might just note, and I haven't actually changed it on the syllabus, I've just changed it on the course website. At some point I'll get around to changing the syllabus and putting up a version 3. But for now, this is the plan. Okay, so if you go to today's material, most important thing, study guide for test 1. So we are not going to cover today's material or last Wednesday's material. I think it's plenty enough to be getting on with to go through 9 lessons. But I have defined exactly what I think you should know from each of the 9 lessons. So don't worry too much about digging into other details. If there are things that I don't ask about on the study guide, I'm not going to ask about it on the test. And if you have, go ahead and skim the list, and if you have questions, bring them to me. Please don't email me on Sunday night. Email me on Friday, email me on Thursday. Do not email me on Sunday night. So if there's something like, oh, this is just really unclear, and even from like your lecture notes or from Google, I can't figure this out, please help me. It might be something that the whole class would like to know too. So if I have any comments, I will post to the Canvas so that everyone can see them. So if you ask me a question, I'll try to share that same information with everybody so that everybody's an even playing field on the test. Yeah, so go through those, and if you have a question about like how much depth of knowledge do I need about this subject, feel free to ask that too. The format of the test is short answer. So I will ask you a question like, why is it important to organize files? Give 3 reasons, and you'll have about this much space, and you'll write down 3 reasons. There's generally not, like, the longest you'll write is like 2 or 3 sentences. It's not going to be essays. It's not going to be, there's no multiple choice. I might have you do some creative things like draw a diagram of how an HPC works, label the head node, the compute nodes, and the queue and the users, or you know, something like that. So I try to get a little bit creative with some of the questions. And I gave an example of scoring and alignment. So go through that, and if that doesn't make sense, let me know. But we talked a lot about alignment scoring, so I want you to be able to actually do that. Yeah, I think the rest of it is pretty self-explanatory. Yeah, so just through lesson 9, don't worry about today, and don't worry about our last class period. Homework 4 is still due on Wednesday. So let me know, say after, if you have questions today or Wednesday. Update on grades. I don't think that Canvas shows this to you, but it shows it to me, so I thought I would share, just so you can see how you're doing kind of in comparison to everyone else. For homework 1, the scores range from 75 to 109 with an average of 96. So I was pretty happy with that. Scores range from 67 to 100 with an average of 86 for homework 2. This is actually with a huge curve. Homework 2 was a disaster. So if you're worried about, you know, oh God, homework 2 did not go well, you are not alone. And I used the curve function on Canvas, which is like super generous. So you guys made out like bandits. So people went from like 30 somethings to an 80. So, but the good news is, like, if you did badly, everybody did pretty badly. I mean, some people did. Some people got like nailed it and got 100s, but a lot of people had trouble with it. So I will post. I have written all the answers to homework 1. I need to go back through and write all the answers to homework 2, and I'll post that. For homework 3, I graded that this morning. Scores range from 60 to 100 with an average of 94. So not too bad on that one either. And yeah, if you guys have any questions about that, or if you're worried about your performance in the course, so now just come and talk to me and let me know. Okay, so we did not finish last class actually getting to variance. How many people were actually able to get to variance? Did anyone try to run it over the weekend? And okay, so not too many. So I'm going to go back to that DNA-seq 3 practical. So I'm going to log into the SSH, to the ACF with SSH. And we'll just let this run while we do the lecture, because it does take it takes like 20 minutes, maybe less. I'm not sure. We'll see. Oh, okay, so once you log in, I think we finally, we have a qsub command that seems to work for everyone. So go copy that qsub command and create a file. I already have it, so, but you're gonna create in your home directory or wherever, but in your home directory is probably best. nanointeractive.sh. I have a bunch of comments. I've been using this file for a while and to qsub for my own projects. But just paste in that one line at the very top. Don't worry about all the pound sign stuff at the bottom. Just paste in this one line at the very top. And then I'm going to save that by Control O and Control X. Now you're going to change mode, user plus execute. And now every time you need to launch that qsub, instead of copying and pasting it, you can just run interactive.sh and it'll qsub for you. And that way you don't have to go back and forth and copy and paste. This is much more convenient than and I also put a little link if you want to just do what I do, which is the SSH ACF and not type the whole like username at tennessee.edu. So I put a little instructions for that. Command line is all about lots of little ways to save yourself time. Oops. Oops. Let's go to the wiki. Okay, so you guys have to help me remember where we left off. We ended up, did we end here? We did the symbolic linking, right? Okay, so if you go to your home directory in the course folder, so put your username right there instead of mine. And then go into 3 BWA and GATK. We have, see, what do we got? We got our intervals that we produce at GATK. We've got our stats file. Does everybody else's look pretty much just like this? Do you guys have extra files, fewer files? Pretty much the same. Oh, that's okay. Don't worry about that. SS, DR. Okay, I think that's, yeah, because I don't have that file. Okay, so back up. We have these 3 directories. We're going to make a new directory called BCF tools. We're going to CD into it. And then I'm going to link to our, the files that we need from the folder, the GATK folder. So I'm going to do BA star because I want to get both the BAM file and that BAI file, which is the index file. So we want both. So if I use a wildcard here and then I do an LS, I'm going to get all of those index files. I think that one's, I don't know, that one probably shouldn't exist. I think these are the 2 you need. But if you copy them all, it should be fine. And then I'm also going to go get our genome file and link that here too. I'll show you. Okay, so remember these are symbolic links, so I'm not copying the file, I'm just creating a little pointer saying, pretend like this file is in this directory, but in reality, it lives somewhere else. So we have our little pointers to all of the files. And if you do an ls-l, it shows you where they're pointing. So it says our dr.bai file is pointing to the real file, which is up one directory and then 3_bwa_an_gatk. So your terminal may also be turning these files a special color. Mine uses turquoise, basically to say, these are pointers. These are not real. These files don't actually live here. They're symbolic links. Okay, so the next thing we want to do is use mpilup to produce a BCF file that contains all of the locations in the genome. This is something that you don't ever really produce an mpilup file. Usually, you just pipe it straight into your variant caller. It kind of just takes that BAM file and it turns it into this very digestible format for a variant calling. So we'll have a pipe in our next command. But first, one of the things that GATK wants to know, or one of the things that BCF Tools wants to know, is the ploidy of your organism. In this case, E. coli, we expect this to be haploid. We expect our variants to be present in every single cell and to only be one copy. If this was a diploid, if you're working on a plant or an animal and it was a diploid, you would want to put, you would want to give the variant caller that information. If it was a hexaploid, you would want to give the variant caller that information because it really changes your statistical model. Okay, so let's create our ploidy.txt file. So I'm going to use Nano and call this file ploidy.txt. I'm going to put *tab *1. So 4 stars and then a 1. You can separate them by spaces. I've always done tabs. I think spaces work as well, but to be on the safe side, you might do tabs because that's what I tested. So what is this? Like, why would we create this file? It turns out that this is an extremely flexible variant caller, and you can specify all sorts of things. Like you can say, especially in insects, this is great. You can say, I'm going to label some of my samples male and some of my samples female, and the males are going to be haploid and the females are diploid. You can say chromosome 1 has 3 copies, but all the other chromosomes are diploid. So all these stars are basically, we're ignoring a bunch of parameters because we don't need them. If you have some complicated system with multiple ploidies and multiple like things going on, you can use these columns to specify that. So you can go into the manual. Daniel? Yeah.
So the number of stars count?
Yes, yes. The number of stars is important. And let me show you this part in the manual. So if we go to the man pages for manual and go to BCF tools, and this is the call command that we're going to use. Here is where it describes the ploidy file. Ploidy definition given as a space tab delimited list of chromosome from to sex ploidy. The sex codes are arbitrary and correspond to the one used by sample file. So basically, you're not going to dig into all of this unless you have some system where your males and females have different ploidy. For us, but the number of columns matter because the columns have definitions. The first column is saying this chromosome from this position to this position for samples with this sex have this ploidy. So for us, we're going to say every chromosome from every base to every base, and for every sex of individual, they're all haploid. So we're just going to put stars in all those positions, but we need exactly 4. But basically, we're saying we don't need to use any of that information. And it does give you an example. The default ploidy can be given using the starred records. Unlisted regions have a ploidy of 2. So if we had not created this file, everything we get, this would be called as a diploid, which would be not right. If you had like a normal diploid where every sample was diploid and every part of the chromosome was diploid, you could just not create this file. All right, so save that. Let me get this out of the way. Don't worry, on the homework, you get to read these files a lot. You'll get very familiar with the manual. Okay, now I'm going to copy and paste this huge command. And I realize that this is pretty unsatisfying because you're like, what the heck is this doing? And that's the first homework question. The first homework question is to break this down, every single flag, every single parameter, and figure out why is it built this way. It probably took me 20 or 30 minutes of reading the manual to construct the command, and I want you guys to go back and figure out why. Why does it look this way? With MPileUp, there's a flag called OU. I'll give you a hint. That's going to have to do with the output, the output format, -F, and we're giving it our reference genome. We have to give it the BAM file. This is creating the MPileUp, and we're going to pipe that straight to the BCF tools software, which is going to use the call function, which is short for call variants. We're giving it some more interesting flags that you guys are going to go look up for the homework. We're giving it our ploidy file, and then we're giving it this output file. Okay, so we're just going to let that run while we go do the assembly lecture. Anybody need me to pause while you get that running, or is everybody feeling pretty good about that? Okay. So de novo genome assembly. This is something that is a little more complicated than just mapping reads. And de novo, the word de novo, or the phrase de novo, is Latin for starting from the beginning. And so when I say de novo assembly, that means starting with raw DNA reads and assembling them into a reference genome. If you're coming from a system where you have a reference genome, lucky you, a lot of hard work has already been done. But if it's your job to construct a reference genome, the first thing you have to do is assemble your reads and try to reconstruct that reference genome. So in an ideal world, you would read an entire chromosome from beginning to end in one long, perfect read. We would have an instrument, or even better, you would have like the Star Trek tricorder. You would just walk up and point it at your plant or your microbe or whatever, and it would produce perfect, amazing genomes that have no errors. That is not reality right now. We all hope it will be reality one day. So the reality is that we have to extract the DNA, many copies of the DNA, break it up into lots and lots and lots of little tiny pieces, and then try to go back and recreate the biological reality using a computer. So there's a lot of steps to go from all these tiny pieces to an assembled genome. And we talked about when you go to NCBI, there are all these levels of assembly. There's complete, there's chromosome, there's scaffold, and there's contig. So some genomes, maybe they have 20 chromosomes, but you actually have 30,000 scaffolds. Why couldn't we put those 30,000 scaffolds back into the right order and orientation to get back to our 20 chromosomes? What is the problem? So I think the most common analogy for this is a jigsaw puzzle, where you're trying to say, how do all of my little tiny pieces fit together? Maybe you have missing pieces, maybe you have dirty pieces, so the pieces aren't right. And it's actually more like doing a jigsaw puzzle when you don't have the final picture. You don't know what it's going to look like until you're done putting all the pieces together. So you don't have that convenient little box with the picture on the front for you to look at. You kind of have to just start as you go. And as genomes get bigger, the pile of pieces gets more and more and more large and complex and difficult to assemble. So I really like this cartoon. Depth versus coverage. Also, often these are synonyms. Actually, hold on, wait a second.
Nick,
yeah?
is there any chance you can upload that version of your PowerPoint?
Yeah, this is the wrong version. That's what I just realized. You guys have the right version. I have the wrong version.
Yeah.
Oh, okay, well, I'll add the cartoon. Somebody reminded me. Oh, okay, we're gonna get there. I rewrote the lecture from last time, and that was the old one, not the new one, because I got rid of all that depth stuff. So as soon as I got to it, I was like, oh, this isn't right. Hold on, I got my folder system is deep. Does that look, that matches, right? Okay, good. Okay, so okay, so the real outline of today's lecture. I'm going to talk about basic short read assembly. I'm going to talk about K-mers and De Bruijn graphs. I'm going to talk a little bit about assessing genome quality. I am not going to have time to get into scaffolding, which is very sad because I care very much about scaffolding, and it's something that I'm doing right now for the Chinese chestnut genome, and I have a lot to say about it. So if anyone else is in that boat and wants to talk about scaffolding, come see me. I'll just mention it at the end. Okay, so we talked about this. So with next generation sequencing, basically you want to start by creating contigs from your very, very short reads and get them as big as you can. And then step 2 is that order and orient your contigs into a superstructure. And that's what's called a scaffold. So the difference between a contig and a scaffold, the contig is what you're generally getting out from your first pass at assembling short reads. After that, when you're trying to put the contigs into relative order, chromosomal order, that's generally called scaffolding. So you can scaffold with mate pairs, you can scaffold with long reads such as PacBio, and you can scaffold with proximity ligation, which is Hi-C, which is what we're doing right now. And it is a game changer. This whole Hi-C proximity ligation, it is going to change the world, because we're taking a genome that we haven't been able to scaffold for 7 years and paid a company $10,000, and they did it in like 6 weeks, and it was amazing. It was like the best $10,000 we've ever spent. So if that's something you need, if you're looking at scaffolding, if you're interested in Hi-C, especially Hi-C as an application for genome assembly or scaffolding, let me know, because I'm super excited about it. But focusing on that initial contig construction, in a kind of similar to the puzzle piece, I think this is a more accurate way to think about it than puzzle pieces. Imagine you had the book Ulysses and you had 100 copies and you put them through a shredder, and then you tried to reconstruct the book Ulysses from the pile of scraps, never knowing what it looks like. And I was asked to give a talk to some beef cattle producers earlier this year and explain to them how genomics works. So I was like, okay, well, what if I find an actual analogy? Like, how big is the cow genome? And it's the size of, like, if you took every book in a medium-sized bookstore, every line of text in a medium-sized bookstore, like thousands of books, that would be the size of the genome. So imagine you took that bookstore and you made 100 copies and you shredded every book in that bookstore, and then here's your room of scraps. Good luck. Go reconstruct that genome. That's the scale of the problem we're talking about here. So we're asking computers to take all these pieces, figure out how do the pieces fit, how do they overlap, are there any missing pieces due to bias, and of course dirty pieces because we have sequencing error and we have real biological variations. We have real heterozygosity, which makes this a big problem. So here is an example. A small genome, friends, Romans, countrymen, lend me your ears. So let's say we had made a bunch of copies and split that up into some reads. And this is what we got back off of our sequencer. You might say, okay, well, what do I see here? I got ROM and that DS. Okay, so those two look like they're overlapping. And RYMEN, RYMAN. Okay, those look like those are probably overlapping. And you would sit there, if I gave you these to you as pieces of papers, and probably eventually, based on all of those overlaps, be able to scaffold them or make a contig. But, you know, and you would have to take into account that there might be some variation. So sometimes, like, most of the overlap looks good, but there's like one letter that doesn't look good. And you just have to be cognizant that that's, you know, you have to be on the lookout for those errors. But maybe if like 5 of the letters match and 1 doesn't, you're still going to call that an okay overlap. And then at the end of the day, you're going to say, okay, consensus. You're back to your original genome. And that is like the most basic example of an assembly that I can give you. Bigger and more complex genomes are more challenging. So difficulties arise from just biological difficulties, which is size. I have a friend who works on conifer genomes that are 30 gigabases. That is 10 times the size of the human genome. They're extraordinarily difficult. they break normal computers. Repetitiveness. A lot of plant genomes have tons of repeats. They're very hard to sequence and assemble. Maybe you have a nice little tiny microbe that only has 25 megabases and it's dense and unique and it's really easy to put together. Maybe you don't. So it just depends a lot on, is the whole genome really big? Is it really repetitive? Polyploidy, of course, is a problem too. I haven't worked a ton on polyploid plants, but people who assemble them are taking on a big challenge. And heterozygosity. So if you're working on a diploid or tetraploid or hexaploid and each copy of the genome is really different from the other copy, then you're just introducing a lot of noise into your assembly. And this was kind of interesting for the peach genome that I worked on a little bit in grad school. We managed to get a plant that was a doubled haploid. So we got the tree, and it actually, somehow, I did not think it would survive. It is still living in Clemson, South Carolina. But that doubled haploid meant that there was no actual heterozygosity in the plant. That is not something you can usually get when you're working in a diploid system. So it's nice if you can get it, but not always possible. On the technical side, sequencing error leads to a lot of chaos with calling bases, and sampling bias, which we talked a little bit about. Illumina doesn't sequence very well through AC or AT-rich regions or GC-rich regions. So if all of a sudden you get to a region in the genome that is particularly AT-rich or GC-rich, you may just not have any reads from that area, and that can cause a problem with assembly. So all of these things kind of contribute to a very computationally intensive problem, and you're not really going to be able to do it on your laptop. To assemble the chestnut genome, we ended up using the monster node on Newton before Newton was the ACF, and that's 1 terabyte of RAM. So that would be like all of our laptops in this room put together times 2 probably would be enough RAM. So you are going to be looking at getting maybe a really, really big computer. Fortunately, we do have that 1 terabyte RAM node here. If you request access from the NSF, you can get 2 and 4 terabyte RAM machines that live in Texas. You have to request access and wait in line to get on them. Again, this is not as big a problem if you're doing something smaller and less complex. But if you're working on some crazy big complex genome and you have trillions or quadrillions of reads, get prepared for that to be a challenge. There's the fun graphic. I think I found a corner piece. That's like when you find a telomere and you're like, we're at one end of the chromosome. We can start going. So volume of information, small genome, 25 megabases, something like that. Might be more like a fungal genome instead of, I think bacteria are generally smaller than that. If you would like to sample each base 100 times, 2.5 billion bases and 25 million reads. This is very doable. Norway spruce is 20 gigabases. They produce 1.9 trillion bases to get this assembled. I think it was still in over 250,000 contigs when they got done. So they didn't get back to chromosomes for sure. But 100x coverage is a reasonable starting point to start trying to do some assemblies. So short repeats are problematic. They are only problematic if they are longer than your read length. So what happens is that if none of your reads can span the repeat and it occurs in multiple parts of the genome, you can't put the pieces back in order. So here's an example. Let's say we have a unique region X, repeat 1, a unique region Y, the second version of the exact same sequence another time, and then unique region C. The assembler, it's going to see that sometimes it has blue and black, so it knows these should be connected. Sometimes it has green and black, it knows these should be connected. And sometimes it has red and black, and it knows those should be connected. So it doesn't really know, because no one read spans all the way through, it can't figure out what goes where. So what it ends up spitting out is your extra deep repeat that's been assembled as like one thing in each of your unique regions. So this is why you don't end up with full length chromosomes, because a priori, you don't know this. What you get back is this, and you have to figure out, is it blue, orange, green, orange, red, or is it green, orange, red, orange, blue, or is it, you can't tell because you don't have any reads that cross that repeat region and can inform you what order these go in. So repeats are a problem. But like I said, they're only a problem if they're longer than your read length. If you have a read that goes all the way through and can prove to you there's blue, orange, green, then you know, okay, there's a repeat in between these two unique pieces and you can figure it out. But if you don't have a read that goes from one end to the other, you can't sort it out. So there are two main approaches for assembly. The old school approach, the kind of approach that was pre-next generation sequencing, and maybe a little bit into the next generation sequencing era, is overlap layout consensus. The example I just gave you of that friends lend me your ear, where we did the overlaps on each read, that is an overlap layout consensus approach. You have lots of little bits, you try to figure out how they overlap, you try to find a path to create your final assembly. It turns out this is too computationally intensive for the size of the problems that we have these days. If you try to run this on a trillion reads, it will break your computer. So we have a new algorithm called the De Bruijn graph. And I'll give you a little bit of an overview of how De Bruijn graphs work. Most assemblers you use these days are De Bruijn graph-based assemblers. So they use something called a K-mer graph. They're designed primarily for short reads. None of this applies to PacBio or Ion Torrent. If you have PacBio and Ion Torrent, I can tell you other workflows to go through. They have their own special workflows that focus more on, they have the long reads, but they have lots of errors. So it's just a completely different type of problem. And De Bruijn graphs are much more accurate, but much more efficient, but a little bit less accurate. So it's a graph-based method, which basically we're abstracting data to nodes and edges. It is a directed graph, so edges may only be traversed in one direction. So it's saying node A goes to node B. So there is a directionality, which we usually represent with an arrow instead of a line. It's a line with an arrowhead, so you know what direction it's going. And the collection of the edges, if you walk along from edge to edge, following their directionality, that creates a path. So the De Bruijn graph it was a mathematical construct long before we hijacked it for bioinformatics use, which is true of almost all of our algorithms. The people who are the best at developing algorithms are people who used to do a lot of really hard math. And they find these mathematical approaches and they say, whoa, that would totally solve this crazy biological problem. And that's how we get a lot of these, which is great. So nodes are subsequences of a longer sequence. And edges are fixed length overlaps. And I think I have a better example of that on the next slide. But basically, the way that we're going to vastly reduce our memory footprint on the computer is to not use the entire read. It sounds like it would use more memory, but it doesn't. You actually break the reads down into little tiny bits, but you only store the bits once. Let me see if that makes sense. So let's say this was your initial segment of DNA, which you don't actually know. This is actually hidden from you if you're doing an assembly. And you get back all of these reads. Now, if you're going to do an overlap layout consensus, you would do this. And of course, for this little tiny example, that would work fine. But let's say you didn't have very much computational memory. What you do is you take each read, let's take this red read, A, C, T, C, G, A, T. We're going to break it down into every possible former. So by former, I mean 4-letter sequence. These are also called strings in computer science. So this would be a 7-mer or a 7-character string. So if you hear me say string, that's what I mean. And we're going to break it down into all of its constituent formers, or 4-character strings. So you have A, C, T, C, C, T, C, G, T, C, G, A, C, G, A, T. So those are all listed here. And then we would do that with each and every read, and we would get all of these little formers. And then what we do is we put all of those formers into a big graph, and the graph essentially does the same thing with the overlap layout consensus, which, bear with me, it looks like it's more computationally intensive, but it's not. So does that kind of make sense? You take your read, you break it down into every possible little word. That's a former. Okay. K-mers, they're also called K-mers, which means you haven't yet defined the length of the word. So we'll see that when you run an assembly, you have to define the parameter K. You're telling it what word length do you want to use? So you say my K equals 28. It's going to go in and subset all of your reads into 28 character pieces. If you say K is equal to 32, it's going to go in and subset all of your reads into little bite-sized 32 character bits. So this is how you start to build the graph. So you take AACC, great. You get ACCG. You say, okay, those overlap by 3 letters, so I'm going to draw an arrow from one to the other. CCGG, those overlap by 3 characters, so I'm going to draw an arrow to the next one. So that's how you get your directionality. As you're reading down the read, you get directionality. Reads with perfect overlaps always have the same path. So in this case, I would have started with this ACC, ACCG. So this is just from the previous slide. You get this graph. But then when you go to the next read, you say CCGG. Hey, I already got that node. I don't have to store it anew. I can use the node I already have. So it goes over and it says, yep, I'm gonna start this read from here. And then CGGT. Okay, I don't have that node yet. I'll make a new node and draw a line. And then my last one, GGTT. I don't have that node yet. I'm gonna add that one and draw a line. So slowly, as you see the same k-mers, you're continuing to build this graph. And eventually you can then walk the path through the graph and get your whole genome sequence. The reads are random, well, you read the reads. So this is an oversimplification because you're actually storing all of these. This would be AACC as well as GGTT. So you're storing the forward and the reverse complement. And you can walk the graph. So you take a read and you start breaking it up, and you look for that k-mer in either the forward or the reverse position at any point in the graph. And then if you found it on the reverse side, you start adding nodes this way. And if you find it in the forward direction, you start adding nodes this way. So because you get both forward and reverse reads from your sequencing, the whole graph is like, can go in either direction, if that makes sense. Yeah.
So is that last part C, is that done by the overlap method, or how's that?
It's the Eulerian path. So you have this huge network construct hanging out in memory on your computer, and then it tries to find the minimum. The Eulerian path, which I think I don't have a slide on this anymore, but I should, is if you visit each and every node if you cross each edge exactly once, each possible edge gets crossed once. So it's going to walk the path, and that's how you're going to get your consensus sequence. It just lives as this giant graph. The reads are gone, poof. Like as the reads get turned into the graph, they get tossed. Map the read into k-mer, shove it into the graph, toss it. Map the read into the graph, toss it. Map the read in the graph, toss it. That's actually what makes it more efficient, even though it doesn't sound like it would. It does, because you're not trying to keep all of your reads in memory. You're just keeping the graph. And then at the end, you have this huge network, and you have to find like the best path through it that makes the most sense. It's a fun, yeah. However, of course, because we have all these problems in sequencing, your graphs get out of control complicated. Like if you have an error at the end of a read, it causes a spur, because all of a sudden your k-mers don't match. So you get a graph that looks like this. If you have a real polymorphism or an error in the middle of the read, this causes a bubble. So your graph does this, and it reconverges. Repeats yield this thing called a frayed rope, because you have, this was that example where we had like the orange repetitive sequence and the yellow and the green and the blue. Because multiple parts of the genome lead into the same repetitive element and then go somewhere else. The reduction, when it's reduced to a graph, it's called a frayed rope, and that's what it looks like. So let's see another example. It was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness. This is a great example because it has lots of repeats. We've got 4 times do we have it was the, we've got the phrase of times twice, we've got the phrase age of twice. So there's actually not a lot of uniqueness to this sequence. And if you completely break it down into a graph, this is the graph you end up with. If you take 5-word, you assume that all of your quote unquote reads are 5 words. Like things go around great until you get to these repetitive areas. And I'm not ever gonna make you draw a graph. Like I'm not ever gonna give you reads or a phrase and make you draw a graph. I just want you to kind of appreciate this like real high level overview of how the software runs. We're gonna run software that does this. It's not gonna tell you it's doing this. You're just gonna say, oh, Abyss, please assemble my reads. And Abyss is gonna say, sure, no problem, here are your contigs. But I just want you to have like a little inkling of like what's going on under the hood. But yeah, so because this has so many repetitive elements, you have these like cycles, and you have weird like bifurcations. And so part of the big challenge of genome assembly is to figure out the most efficient and quote unquote correct way to walk this path to produce this. So you don't actually know this. This is what the computer is starting with, and it says, huh, what's the most likely genome to produce this? Yeah.
So I'm assuming you can play with the K value?
Yes.
So how does that enter into all of this? Like, how do you make a decision about these? Okay.
K-mer size is important. And this is a great paper looking at E. coli and assembling it, and they just did different K-mer values. A K-mer of 50 produced this graph, and you can see like lots of nodes you enter, and they're collapsing. Anything that's unique and isn't duplicated got collapsed down into a circle that was like this. The circles are basically something that you can walk the path super easily. And there was any like confusion or inability to figure out how to walk the path, they left it in the graph. So you can see like there's still a lot of chaos. Like putting the E. coli genome back together turns out to still be really hard with a K of 50. With a K-mer of 1,000, you have a much simpler graph. There's a lot less chaos. You can figure out how to put all the pieces in the right order and where the repetitive elements are a lot more easily. With a K-mer of 5,000, it assembles beautifully as one perfect genome. This is assuming that you don't have any errors. So the trade-off with K-mers is that a shorter K-mer, it builds a much bigger graph, but it's a lot more accurate. Because if you have, what's a good way to explain this? So every time there's an error in a K-mer, and you get like a bubble, or you get like a frayed end of a loop, if you have short K-mers, it's easier, it's called popping the bubble, to like collapse your polymorphisms or and like with a frayed rope, the smaller K-mers can be a lot more accurate, but computationally way more expensive. Long K-mers will give you this much, if it was entirely accurate, it would be better, but it's harder to fix errors with long K-mers, and they're less computationally intensive because your graph is much smaller. So it is a trade-off. What we're gonna see, what most people do with like Abyss is like run it with a whole bunch of different K-mer values and see which one works best. Keep running it until you run out of computational ability. And usually, in general, bigger K-mers are better. You're gonna get more accurate results. But you will eventually run out of computational power and not be able to. And your K-mer can't get bigger than your read, right? The biggest your K-mer can be, they generally suggest, you know, like 50% of your read length is a good place to start with your K-mers. And of course, it's computationally impossible for it to be longer than your read length. I loved this blog. This was great. This was a metagenomics artifact. And they like wrote the software to actually like look at the graphs that were being produced. And I just was like, that is so ridiculous. So like we've had these kind of nice small examples with these little poems, but in general, what your computer is actually chewing on is something like this. And when your assembly falls apart and you have whole regions that you get nothing back, or if you have major misassemblies, it's because the computer encountered something like this and said, I have no idea, you're on your own, right? Like this is the type of thing, like sometimes because genomes they're not perfectly random. You have these repetitive elements, you have these things that cause all this chaos in the graph, and it just turns out to be a huge problem. So I guess if you used a K-mer that was exactly the length of your reads, that would be almost an overlap layout consensus method. Almost. It'd be close. It would still use the graph, but yeah. Smaller K-mers require less memory, but the graphs are more complex and they yield smaller average config lengths. Repeats longer than the K-mer length cannot be resolved. So that's a huge problem. Larger K-mers require much more memory. but they can yield longer contigs. They yield, paradoxically, they yield both longer contigs and also lots of small contigs that are actually little overlapping pieces that it couldn't assemble because the K-mer was too long and there was an error in there and it couldn't figure out how to collapse them. So larger K-mers are usually better, but not always. So you usually want to try a set of K-mer values and pick the best. Start with 1/2 to 2/3 the length of your read if you have sufficient RAM resources, and then maybe try a few higher than that and a few lower than that and see where you end up. Ben Langmead wrote the Bowtie program, and he's a very, very, very good mathematician and bioinformaticist, and he does this great YouTube series that will do like an $80 million better job than me at explaining this. Like when he breaks it down, it is like so simple and clear. So I feel like I do a poor job of explaining these things. They're complicated, they're difficult. I don't expect you guys to be able to regurgitate them to me. But if this is something you're interested in, if you're kind of a mathy person, if you're interested in algorithmic development, I don't get a lot of students like that, but if you're one of them, please go watch these videos. One of the classes I wish we had at UT was a bioinformatics algorithms class, and we don't. So if you wish you had that class, you can just follow Ben Langmead has all of his lectures online, and they're amazing. They really dig down into like how the math works and how the algorithms build these graphs, and he does a lot with read mapping, and it's really good. So check that out. Some programs that I recommend for big eukaryotic complex genomes, a lot of people are using AllPaths. It's common. It requires both a mate pair and a paired-in library. So if you don't have a mate pair library, that could be problematic. And it's pretty clever at managing memory. So you can get a lot bigger assemblies done with a lot fewer computational resources. And I think that's one of the reasons people really like it. If you're working on bacteria, Spades and Abyss have been around for a long time and do a really good job. Spades has been updated to do metagenomic assemblies and is doing a pretty good job at that. I think there are a number of options in that field, but I know Spades is one of them. Single cell sequencing, Spades and Abyss both have updates to support single cell sequencing. And they both, and I think all three of these have proper utilization of paired-in data in the De Bruijn graph. And that's sort of a new thing. The original De Bruijn graphs only used the single ends to build the graph. Even if there were paired-in data, the relative location of the two ends wasn't actually being used, which was just like a loss of a huge amount of information. But all of these actually do properly utilize the paired-in data, which is great. Okay, so assessing genome quality. Contiguity, that's a good first step. You would like fewer contigs and longer pieces. That's the goal. And I mentioned this, so you'll generally in your paper report, these are the number of contigs or scaffolds. The average length is this, and the N50. The N50 is something that you don't see anywhere else except in reference genome world. So what is the N50? So the short answer is 50% of the genome is in contigs as large as the N50 value, which is not super intuitive, but that's what it means. So if you take this genome that is 1,000 base pairs, and we got all of these contigs ranging in size from 300 base pairs to say like 5, right at the 50, and we order all of the contigs from largest to smallest, we go to the 50% mark, and the 50% mark is right on this contig with 30 bases. The N50 is actually, well, it would be 45, because if it's halfway through a contig, you have to go up to the 45. 45 bases would be your N50. So again, the length through which the collection of all contigs of that length or longer contains at least 50%. Similar to a mean or a median, but it makes your assembly look a little better. It's always going to be bigger than your mean or your median, because basically what you're trying to say is I don't want to weight all of these little tiny guys at my 50% mark. I want to pretend like they don't exist so I can get into nature genetics. No, it's a statistic that just, it tells you like half of my assembly is in pieces this big. So it makes your assembly look better, right? Does that kind of make sense? It's a really weird way to do like a median mark, yeah. At least 50% of the whole assembly length is in contigs of this size or bigger.
You need to specify that size.
Yes. So my N50 for this would be 45 bases, because half of my assembly is in pieces of at least 45 bases or bigger. Or no, I might be wrong. I think I have it pointing at 30. That is at 30 or bigger, even though the 50% marks happens right in the middle of the 30. Yeah, so 50% of my assembly is in contigs of at least size 30. So if you were randomly looking for a gene, there's a 50% chance it would occur on a contig of at least 30 bases. Okay, so a couple of examples. Yeah, Daniel.
So what they're always would be one-on-one assembly.
Yeah, so this is generally not used for something that's in a chromosome level scale of assembly. So soybean would just report we have 95% of the chromosomes reconstructed correctly, and they would move on. But if it were still in like thousands of contigs, you report the N50. That's a good point. If you're up to like chromosome level assemblies, you don't worry about the N50. But if you have 10,000 contigs and you want to say, well, my N50 is a megabase. I don't know. It just is. It's a metric that you hear a lot that most people don't know what it is. So I think it's worth teaching, because most people are like, what the heck's an N50? Rightly so, because no one else in the world uses it that I know of except reference genome people. So does that make sense? Okay. So assembly A contains 6 contigs, so 80, 70, 50, 40, 30, and 20 kilobase pairs. The sum size of assembly A is 290. So if you added all of those up, that would be 290. So half of 290 is 145. To get up to 145, that would be 80 plus 70, already gets you there. So your N50 is 70. So half of my total assembly size is contained in these 2 contigs. So my N50 is 70. Assembly B, another example, 80, 70, 50, 40, 30, 20, 10, 5. If you get a calculator out, you can sum all of that up to 305. Half of 305 is 152.5. This gets you to 150. You have to actually add one more contig to get to your 50% mark. So for this one, the N50 contig length is 50 kbp. Math, I know. That's how you calculate it. Okay, so you're always going to see if it's not chromosome level, you're always going to see number of scaffolds, average length, and the N50 length. And the N50 is always going to be way higher than the average, because that's how it's designed. It's designed to kind of make the assembly look better, to focus on the 50% that's well assembled and ignore the 50% that's very badly assembled, if it's badly assembled. So completeness, another way to assess genome quality is it complete? You might want to ask how much of the total genome was assembled, 80% complete versus 99% complete. This is kind of based on your understanding of actual genome size. First of all, do you actually know the size of your genome? But the other thing you can ask here is about genes. How many of my genes did I capture? Most people are more concerned with this than they are with the whole genome. Because a lot of those repetitive regions are difficult to assemble and maybe not that important. So how many of my genes were captured in the assembly? You can look at the percent of your RNA-seq reads that map to the genome. If 99% of them map, you're in good shape. If 42% of them map, you probably have a lot of missing genes. The other way to do this is BUSCO, which is conserved core single copy orthologs. And they have a BUSCO database for plants, and they have a BUSCO database for arthropods, and they have a BUSCO database for mammals. And so they just have this huge group of what they think are conserved single copy orthologs, and you can go look for them in your genome and say, as a proxy, you know, I captured 90% of the conserved single copy orthologs, therefore I think my genome is 90% complete. You can also do some fun ways to try to look at correctness. So how error prone, how bad is my assembly? Is it good, is it accurate, or do I have problems? So what you'd be worried about are misjoins, collapsed repeats, miscalled bases, insertions, deletions. Reference genomes, especially 5 to 10 years ago, were rife with real problems. Like taking the reference genome as the gold standard is not always the best idea. But you can assess this by aligning all reads back to the assembly and looking for inconsistencies. So if your paired ends are mapping to vastly different places, that may indicate a misassembly. Or if a lot of reads are mapping and showing you that, you know, that base doesn't really exist, very well it could be that that base doesn't really exist. A program that you can use for this is called Quast. I will compare assembly attempts to each other, so that's really nice. So if you're running your K-mer at 5 different K values and you want to compare them all, Quast will do that. It'll give you some basic statistics about contigs and scaffolds. And it will map your reads back and tell you the % of the reference genome covered by the assembly. So that's pretty nice. Yes, so I will not be talking too much about this, just a little bit. Most of our programs that do assemblies will do some level of scaffolding. It will use paired ends and or mate pairs to go ahead and try to do some scaffolding. My current favorite thing in the world is this proximity ligation technology. I've been working with Dovetail, the company, or my collaborators have been. The other one is Phase Genomics. There might be others out there that I'm not aware of. They do this amazing job using high-C data to scaffold assemblies. You can also use PacBio. There's nice long reads to figure out how your short contigs should be ordered. Optical mapping is something I don't know a lot about, but I hear good things. And of course, linkage maps. Good old traditional genetic linkage maps are really helpful for putting things in order. So some combination of those. Lots of people end up using 2 or 3 or 4 of them. And I think I mentioned this, but I want to be clear. What is a scaffold versus a contig? So the contig is when you generally know every single base, and your short reads have produced this. Your scaffold is more where you don't know what's in between your contigs, but you know what order and orientation they go in. You know, this one comes first, and this one comes second, and this one comes third. You may or may not have much of an idea what goes in between. So you generally just fill it in with a bunch of ends. So in the middle of you like paste your first scaffold sequence and you say in. And that just means, I don't know what goes in here. Something goes here and I don't know what. And then you paste in the next contig and then in. And if you go to genomes, like these still exist in the human genome and the chicken genome and the soybean genome, you'll find these huge gaps of ends where like, we still don't know what's in here. For whatever reason, this region won't sequence or we can't figure it out. Yep. So some people just say, I have no idea, I'm gonna put 200 and call it a day. So all your gaps are exactly 200. Sometimes they try to use the size of the paired end or the mate pair fragments to estimate. And say, I think these are supposed to be 10 kb apart. To make this gap exactly 10 kb, I have to put in 8,000 ends. So you have to read the genome paper to see which strategy has been taken. Yeah. If you have something like PacBio, where you're putting things against the superstructure, you generally know about how far apart they are. But if you're just using mate pairs or pairs ends, it's a little fuzzier. Yep, and so you basically, once you have all this information, you make a new FASTA file with your nice long scaffold sequences. Here's just another way to look at this. So oftentimes your short paired end reads are gonna really help you build your contigs, but your mate pairs that are like 10, 20 kb apart are what really help you to put those contigs in order. But if you don't know what goes in here, that's where you stick the ends. And you can either stick 200 ends or you can like try to figure out what a likely gap size is. And a lot of this used to be done with physical maps, but no one can afford to make physical maps anymore, so. All right. Any questions? Assembly is hard. It's the hardest thing, for sure, I think. Okay, so this has probably been going for about an hour, so I'm gonna actually log out and I'm gonna exit this Q sub and re-Q sub because it'll just die in the middle of our lesson because we only get an hour. So it gives you a nice little message, your Q sub job is complete, and now I can just go to my interactive script and it'll launch a new one without me having to copy and paste, which makes me happy. Well, hopefully it launches eventually. Okay. All right, so now I'm going to go ahead and get back to my directory. It always pops you back into your home directory when you re-QSub. So you have to CD back to your project directory. So you will do that except use your username instead of mine. And now hopefully everybody has this vcf.gz file. And VCFs, even though they're gzip, they're pretty human readable. Like, okay, if I do a head on the spam file, I bet it's going to be a total disaster. Like, that is binary, you can't read it. But VCF files, even if they're gzipped, let's see what it looks like. Oops. No, maybe not. Okay, unzip it. Oh, and don't type VI, sorry. If you get stuck in, I don't think I did. If you get stuck in VI, raise your hand and Rula can come help you get out. I just type it like automatically. Okay, so we have a VCF file, yay.
You might do a word, mine would just like middle of it.
179 lines, it's pretty short. I know. Okay, let's go look at it. Okay, so the header always starts with the pound sign. It's telling us that it's a 4.2 version of VCF. It gives us the actual command that we use to produce the VCF, and it gives us a bunch of information. If we scroll down, all the way down, those are our actual variants. So it's saying your chromosome at position 8,698 has a variant. The reference is, this must be, yes. Oh, it does not have an ID because we did not give it any known SNPs. The reference allele is an A, the alternate allele is a G. So in comparison to the reference genome for E. coli, our sample had a G instead of an A at this location. Quality value of 228 is really good. No filtering has been done yet. Our read depth was 250. I can't remember what BDP means. We could go look these up. Lots and lots and lots and lots of information. If you go all the way to the end, it's going to tell you for each individual sample, we're going to give you the genotype. And then I think this is a probability score. So the genotype is a 1. So it's the alternate allele. If this were a diploid, it would say 0/1 or 1/1 or 0/0. But since we told it was haploid, it's only giving us one. It can only be one thing. And the probability is very strongly on the side of this being the alternate and not the reference allele. And if we had more samples, they would appear as additional columns out to the end. But since we have one sample, we only have one set of information about the sample. So if none of that made any sense, you might want to go look at the VCF lecture from last week. Here's an indel where it's saying, okay, the reference is ATT, the alternate is AT. So basically we're missing a T. So this was a deletion of one base in our strain that we looked at. You can go down. Here's another one. The reference had 5 Gs. Our strain only has 4 Gs. And then the rest are pretty much SNPs. So those are your variants. These always kind of worry me. Like, I feel like it's hard. Like maybe the sequencer had a hard time calling enough because it was T, 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 T. Although those types of runs in the genome can also be really polymorphic. How do you check if this is real? I mean, it's definitely a repeated T. I mean, that's obvious.
I mean, if you may have the same rate of EPPs, but
So if you have a good reference genome and that reference genome had those locations accurately represented, you would have discarded your multi-mapping reads. So what I would do is you map the reads and you only keep reads that map uniquely. If the reads map to multiple locations, you don't want to use them for variant calling for that exact reason. So you remember we talked about way back when we were doing mapping, you have to decide, do you want to keep your reads that map to multiple locations or not? If you're doing variant calling, you probably don't. Because how do you know? How do you know if it's just a variation between the two repeats versus a variation within the same location? So if you always throw away your reads and map to multiple locations, that helps. If your reference genome isn't right, if that repeat has been collapsed and is only represented once in the reference genome, that could be a problem. For that, we can do depth filtering. So we can say anything with less than 10 reads, throw it out, because I didn't sample it adequately. Anything with more than, let's say you know that your average depth was 100. If this region has 300 depth, average depth, maybe that's actually a repeat. Like that's way out of whack with what my coverage should be. So you might want to filter for depth if it's really extremely deep, as well as if it's really shallow. There's only two ways I know of to do it. Go in and Sanger sequence a slightly larger region. If your repeat is smaller than a Sanger sequence, then you can sometimes sort out if you have multiple regions. But of course, then you have to have primer. You have to know enough about the region to design primers. So it's a tough question. Speaking of filtering, the next thing we can do is filter. Oh, let's do stats first. So BCF Tools comes with this great stats program. So I'll type it out for you. BCF Tools, bin BCF Tools. Stats. So we are going to give it our reference genome. We give it -s- and then our BCF. And I might have to gzip that again. We'll see. It's already unzipped it. BCF stats. So this is a just a little statistics, little breakdown of what you have in your file. It's going to tell you basic information. You had one sample, 150 different records, 138 of those are SNPs, 12 of them are indels, no multi-allelic sites, haploids, so that's good. And I have to look up what all of this means. I usually just use this little section. But if you break all of this down, it gives you a lot more information about like your transitions versus transversions, indel, context. So repeat inconsistent. All of our indels are repeat inconsistent, which we noticed. It was like always the TTTT that, you know, we had 5 T's instead of 6 T's or 6 T's instead of 7 T's. So they were all repeat inconsistent. Yeah, so just a whole bunch of information. But the super basic stats are always at the top, which is here. One sample, 150 variants, 138 are SNPs, 12 are indels. Okay, so at this point we can now filter. And I'm just gonna filter for anything that's low quality and keep only things that are quality value above 10. Oops. And we don't have the .gz anymore. And then, so your job, I think for the homework, it asks you to run that stats program on this file and give me the information about how many indels were filtered out and how many SNPs were filtered out. So it's basically just to take the previous command and run it on this new file instead of on the old file. Also, you can also, and another thing you can do, and I think the homework asks you to do this, is open up your variant file and find one of those variants and make sure it looks real. Are the variants where you expect them based on your VCF file? Make sure it looks real. You can use SamTools TView for that. Okay, so all of that is from the material from the last class period. So work on that, but also we're gonna start on our assembly challenge. So, the most frustrating thing about this class, and I understand it, is that you feel like you're just copying and pasting commands, and like this first part of the class, it's really hard to put together, like, what am I doing? Why am I doing this? What all these commands mean? So we're solving that this week with two things. Your homework is asking you to parse these commands and understand the flags and figure out, that's gonna hopefully get you thinking about, if I had to write this command from scratch, could I do it? The second thing we're gonna do is a lab where I don't give you any of the command lines. I'm just gonna give you this umbrella of like, this is what you're gonna do. Go forth and do it. So your challenge is to figure out how to run an assembly. As a class, we are going to test out different K-mer values and see how they impact our assembly. So read the documentation and run a BIS on our trimmed E. coli reads using your assigned K-mer value, which if you click on that, it should pop you into a little Google Doc. And each and every one of you has a little K-mer size that you're gonna use. And we have 3 replicates of each K-mer, so we can see accuracy. If there's someone who's a total outlier from everyone else, we will know. So go look up your K-mer value. You will then need to get some statistics on your assembly, such as the N50 and the number of reads and the assembly, like the average contig length. And you're gonna fill in the spreadsheet with your assembly metrics. 2 extra credit points on a homework if you can complete the challenge and get the answer pretty close to what I got. I'm gonna have to figure out some way that you can show me. I'll probably get you to like send me the FAFSA file so I can see you just didn't copy the numbers from someone else who had the same K-mer. I mean, I know none of you would do that, but just to be on the safe side. Installing a BIS is a little bit of a pain, I'll admit it. So I went ahead and did that for you. So here is the location of a BIS. If you look in that folder, there's a whole bunch of different options. You have to read this documentation and figure out which one of these BIS programs are you supposed to be using. Use the trimmed paired in reads from our E. coli example. So use the same E. coli files that we've already used and use the ones that we trimmed. And I have a little software program installed that gives you the assembly stats, and the usage documentation is here. So I'm pretty sure you guys won't complete this in the next 17 minutes. So we'll be doing this for today and on Wednesday. All right, so enjoy. I'm gonna let you guys just chew on it for a while and raise your hand as you have questions.